This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 227, recorded on April 5th, 2013. Hello, everyone. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today right here in the TWiV studio, Dickson Despommier.
Hello, Vincent.
How are you, Dickson?
Chipper. Feel real good.
Chipper.
And not in the Fargo sense of that word, by the way.
I don't know what that means.
You would if you saw the movie.
I didn't.
When you talk about Chipper in the movie Fargo, you'll see what I mean.
Okay, but I'm never going to see it.
No, don't do it.
Do you like the movie?
I love that movie.
I'll never see it then.
That's a genre movie. It's really fun.
Okay. Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
How are your pigeons, Alan?
Oh, they have taken flight, I think.
Alan, are doves positive for influenza?
I don't know. Well, pigeons are a type of dove, so...
That's true.
They are?
Yeah.
Huh. So,
yeah, they're sometimes called rock doves.
Wow.
Yes.
If you had another child, you could call it Rock.
Right. Actually, there were a number of child names that we couldn't really consider because of the last name Dove.
Interesting.
Lonesome would have been a good one.
Yeah.
Also joining us today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everyone.
How's Ann Arbor?
It's good. We have a nice sunny day, 44 degrees.
Any daffodils?
Not yet. Way too early for that this year, anyway.
Right.
We have them.
Yeah, we do.
We do. We have daffodils, crocus.
Yeah, but we've got a heat dome over the city, though. That doesn't count.
We have. It's very warm here. It's 11 degrees Celsius.
Right.
And it's blue skies, and we have a red flag warning on the weather forecast because of the enhanced threat for spread of wildfires.
Yep, same here.
It's crazy.
Why?
Isn't it?
You're probably pretty safe in Manhattan.
No rain.
But no rain and high wind. People think of wildfires, they think of heat, but really what you need is dry tinder. You can have a wildfire in the middle of the winter.
Alan, you don't have any snow up by you, do you?
It's almost all gone. The foothills of Mount Snowplow are still scattered around town in a couple of parking lots, but other than that, it's almost all gone.
Yeah, we don't have any here.
Well, earlier this week it was really cold here, so this is
True.
a nice turn of events.
Definitely
true.
I hope it stays like this.
And no wildfires.
No wildfires, no. All right. Today we have kind of a potpourri or a potpourri of stories which have been in the news, and I thought we should talk about to get everyone's take on it. Dixon, you got your iPad working there?
I have a take. Yeah, I'm reading along with you.
You got your email working?
I do.
I can't believe it.
No, come on.
You actually remembered your password.
Stop! I didn't have to. It just came up.
Oh, that's why. I just have to let everyone know I have to keep Dixon's password because he forgets them all the time.
Well, if I wrote them down, then I wouldn't forget them. But that's the way it is.
We have a follow-up. I received a tweet this week from Paul Dupre, who I saw last week in Manchester, by the way. Hello, Paul. The tweet says, in Threading the Needle, you discuss polio becoming BSL-4 when eradicated. Do you have a reference for this? Very interesting news. So I looked very hard, and I found a document called Global Action Plan, WHO, and in it they talk about the containment that will be necessary after polio is eradicated. So there will be a period where it's BSL-3. So after the last reported case, for a while it will be BSL-3. And then when it's truly eradicated, then it will become BSL-4, and I believe it's documented in this document. And of course, they really would like people to destroy all of their stocks. That's the idea. They don't want a lot of people to have polio anymore. And then whatever is left, if you have to work on it, you do it in a BSL-4.
Good luck with that.
Right.
There's also a website which is very good, which I think we've mentioned before on Twitter. It's called Polio Eradication Initiative, polioeradication.org. And there is a page full of post-eradication documents. and I'll put a link to that in the show notes as well. They talk about cessation of vaccination, developing affordable inactivated vaccine. Have we talked about the new stance of WHO on adding a dose of IPV to immunization? I don't think we have we?
I don't think we've talked about it on the show.
So WHO now, his official policy is, Alan and I said in 1997, you need to switch to IPV.
Right.
You can't just stop using OPV.
Right, because for the refresher for people who may be new to this topic, OPV is the live viral vaccine, and probably everybody who takes it sheds virulent revertants in their stool. And that's the backbone of the eradication campaign in developing countries because it's a very easy to administer vaccine and very cheap.
Yeah, and so if you stop immunizing, you have all new susceptibles, and they can be infected by circulating vaccine-derived viruses. And we know this because there have been outbreaks in various countries caused by those viruses when immunization levels drop.
So you have to switch to the inactivated vaccine until all of your vaccinees have stopped secreting, which, given that a lot of immunosuppressed people have now received the live vaccine in these eradication campaigns, it's going to be years.
And Alan and I said in 1997, this is what you should do. And WHO replied, no, it's not necessary. And so this year, they finally said, yes, we have to switch to IPV. It's quite clear. And so they said, WHO, it's a very interesting position. They said all countries should immediately introduce a single dose of IPV before the OPV series. And they're not telling the countries how they're going to do it because that has to be injected.
Yeah. And
it's much harder to do that than to just put a few drops. I mean, even Bill Gates can put a few drops of OPV into a child's mouth. But injecting is another matter.
Well, he's good at giving people viruses.
That's great.
Excellent.
Hey, Bill, Bill, you should listen to this show.
Yes.
And you should support it, too.
It's available on the Zoom.
He might listen to it. You don't know.
He might.
How would I know?
You wouldn't unless he wrote you a letter.
So the idea is that IPV will begin to provide immunity, and WHO says then when it looks good that there's no more type 2 around. Right now there's no wild type 2 polio, but there is circulating vaccine-derived polio. So they're going to stop giving type 2 OPV, just do 1 and 3, and have this IPV. So when circulating type 2 is gone, then we'll stop giving, no, they're not giving type 2 OPV, or maybe in some parts of the world they still are. They'll stop using type 2 OPV, and then they'll follow the same plan for types 1 and 3.
So, Vince, I have a question, and I asked this before, too. When I first heard about this, we were just discussing this in private. The longevity of poliovirus wild type outdoors is how long again?
It really depends. There are some hard numbers, which I found this week, actually. I should have saved the link. It has a half-life, which would suggest no more than a few months in the environment.
Okay, so in the areas which are considered now to be endemic, which would then be eradicated by IPV, followed by an OPV regimen, let's say, couldn't you then survey the effluent from those places and then look for those viruses? When it's dead, you've eliminated it.
Well, recently they've found in the sewage in Egypt poliovirus.
Abroad, of course.
And there was no paralytic disease there, but they have surveillance, right? But you can't look at everything. So your conclusion is only as good as your surveillance, and they're going to miss something. So you have to really be certain.
And empirically, when the eradication campaign leaves a particular country, the surveillance tends to become a lot more lax.
Correct. But this still begs the question of how are you going to look for the virus, and you need a laboratory, and you need to isolate it, and therefore you need to grow it. But this other regulation about destroying all the viruses is not going to work then, because you're going to have constant testing going on, and sometimes
you don't need
you'll
the virus to test for it.
No, but sometimes you'll find it, though, because it will grow in the
that's a conundrum. Yes, when you do find it, you have to destroy it right away.
Yes, exactly right.
That's right.
Duh.
But there are many, many regional laboratories. In fact, our friend Jason down in Australia his lab is one of the regional WHO laboratories where they do surveillance for WHO. So this is good that they're switching to IPV, and the thing is that not a lot of production capability at this moment. I think Sanofi Pasteur makes the most IPV of any country.
Right.
And other countries are going to want to make IPV. They should, like China and India probably. But right now, IPV is produced using virulent strains of polio.
Right.
All right. So you have to be really careful. And if polio becomes BSL-4, that's going to be hard to make a vaccine in a BSL-4. I mean, Alan and I could barely move around in it.
Exactly.
And I can't even fathom, Boston University spent the federal government spent $200 million to build that building in Boston that we toured.
Yeah.
Right.
Now, imagine a pharmaceutical factory
No way.
that has to be inspected on a regular basis and has to meet GMP and all these other requirements, and now build that inside a BSL-4. It's the cost of that is going to be insane, and frankly, I don't think anybody's going to fund it for a disease that's regarded as gone.
Now, some solutions would be to have a non-infectious vaccine. And in fact, the Gates Foundation is supporting research into alternate polio vaccines. I mean, we're going to have to stockpile one forever in case of an outbreak. And one of them would be an empty capsid vaccine. I talked to some people in Manchester last week. They're working on that. Another idea is to use the Sabin strains to make IPV. They are less transmissible and less virulent. So it's not clear what the post-eradication vaccine will be yet, but now that it's getting closer, WHO is starting to pay more attention to it.
Right.
It'll be interesting to see what comes out of that. I mean, Eckhard Wimmer had made a virus where he changed all the codons in the capsid region so that they'd encode the same amino acids, but the RNA was totally different. And that virus is attenuated, and theoretically it doesn't revert because there are hundreds and hundreds of changes. So that's another possibility. He's working with a company on that. So this will be interesting to see. It's curious now. We can eradicate the disease with OPV, but we have to switch to another vaccine afterwards.
And developing a new vaccine, of course, the window to do that is closing because you're not going to have endemic areas where you can test it anymore.
Yeah, so actually I talked with Jeff Almond about that. He's a virologist at Sanofi Pasteur, and he said you will just have to show immunogenicity.
It'll be like a lot of these other select agent vaccines, but then you're taking a proven time-tested vaccine and replacing it with a stockpile of an untested, essentially, vaccine, and hoping that it works when you really, really, really need it.
Yeah, it's iffy, but I don't know what else you can do. The other thing that I find...
Well, I know what else you could do. You could set aside the concept of eradication and say, let's simply bolster public health and immunization against all of the vaccine-preventable diseases.
True. Oh, that.
But that doesn't win anybody any Nobel Prizes, so
no.
It's a non-starter.
Kathy.
What I was going to say, back when you were talking about everybody destroying all their stocks, it seems to me that people places might have frozen stool samples and things too.
Sure.
And I don't know if you, Vincent, kept track of everybody that you sent your infectious clone to, or if they kept track of who they sent it to, but people just, potentially have those plasmid DNAs sitting in their freezers.
Absolutely.
Yep.
Absolutely. That's why we need to have a stockpiled vaccine, because you may most likely get an outbreak at some point.
I think the solution is to continue vaccinating.
Well, I'm sure the U.S. and Europe will with IPV for many, many years.
Yeah, and in fact, my daughter got IPV as part of one of these massive multiplexed vaccines. And I forget what it was. It's a whole series of things, and IPV is in there, and that's just one of the shots. And that's the way to do it, is to get that to the whole world. And, because even if we do eradicate polio, then you've still got kids going blind from measles, and you've got mumps.
I mean, the other thing I worry about is having an injectable vaccine, because it's really more expensive and it's harder to give to many people. And we've talked on TWiV about these microneedle patches, and I would hope someone tries to adapt polio vaccine to that. That would really help a lot, right?
Yes. Mm-hmm.
It's a really interesting topic, though.
It is.
Anyway. So.
Yeah, it's a tricky problem.
Can I just ask a naive question? Of all of my questions are naive, but this is particularly naive. I should know the answer to this already, but what is the next closest virus to poliovirus?
So there are a number of enteroviruses that are quite close, Coxsackie viruses and other enteroviruses, And why you think they might jump in?
No, I didn't think that at all. I just wondered why this is the only one that only infects people and not anything else. And,
actually, there's a paper on this which we should discuss another time about the closest relatives and potential for other viruses taking its place.
It makes you wonder where it came from. Where it came from. That's all.
Yeah.
Well, that's a really cool question. We don't, for some viruses we have some inkling of where it came from, what animal and when, but polio, there's just no clue.
That's odd, isn't it?
Well, it's not, someday we will find a closely related virus in an animal somewhere, but we don't really look.
Hopefully not by accident.
Right, exactly right. That's
So
the
that
carrier. Oh my God.
That makes me think about the smallpox story and the emergence of monkeypox.
Yeah.
Because there was cross protection from the smallpox vaccine.
Right.
There might be protection against something zoonotic that we don't know about.
Yeah.
Sure.
Well, I was thinking of the coronaviruses, actually.
Well, I tell you, if that happens, I still have a lab, I'm going to jump right on that, on that virus.
Yeah.
The coronavirus people, they jump all over these
they
new
do.
ones.
They do.
The flu people are jumping
right.
all over. And I'm just waiting for my virus. And I hope it doesn't happen.
Yeah.
All right. We have a couple of follow-ups about getting flu vaccine in your eye.
Right.
Kathy, could you read the letter from Hannah?
Sure.
Hannah and her sisters.
No, just Hannah. Right. Dear TWiV, on episode 218, Welkin Johnson recounted how the syringe broke as he got his flu shot, and some of the vaccine ended up in the eye of his daughter. He asked whether this might have had any effect on her immunity. You said probably not, given that it was an inactivated vaccine. I agree. You then wondered more generally whether it would be possible to get infected this way with a live virus. Then, last episode, 225, you read a letter from a listener pointing to some studies of influenza infection following ocular infection of ferrets. You wondered how the virus got to the respiratory tract and whether this would happen in humans. In short, Kathy is not totally wacko. The eyes and nose are indeed connected. I'd suggest this helpful review of the topic written by some of the same folks. As the authors put it, the nasolacrimal system, which provides an anatomical bridge between ocular and respiratory tissues. The lacrimal duct collects tear fluid from the ocular surface and transports it to the inferior meatus of the nose, facilitating the drainage of virus from ocular to respiratory tract tissues in a replication-independent manner, thus serving as a conduit for virus-containing fluid exchange between these sites. The article also notes that ocular tissues have the cellular receptors for many respiratory viruses, meaning that in many cases, virus can replicate in the eyes. Think about that, or try not to, next time you rub your eyes.
Dear.
So,
It's a great image in that paper.
yeah.
They have.
This is the one by Jessica Belser, Paul Rhoda, and Terrence Tumpy. Yes. That is a good one.
Ocular tropism of respiratory viruses. So there's this figure 1, picture of a human, and showing all these connections between the upper tract and the eye. And the cool thing, I think, in this figure is they show you what kinds of sialic acids are at each site.
Mm-hmm.
Alpha-2,6 being what human influenza viruses prefer. But the cornea and the conjunctiva, as well as the nasolacrimal duct, have alpha-2,3s. Mm-hmm. Implying that they could be infected by avian influenza viruses. Pharynx alpha-2,6, larynx alpha-2,6, trachea alpha-2,6 and alpha-2,3, alveoli alpha-2,3, bronchial, etc., or a mixture. It's a cool figure.
Mm-hmm.
Very nice. Why don't you read Trudy's as well? It's related.
Okay, Trudy. Hi, Twivers. I'm writing this email as a follow-up to my email, which was read on TWiV 225. In that email, I brought up the possibility of flu infection through the mucosa of the eye and forwarded you a couple of papers about this topic. Since you brought up the question of how the virus could end up in the respiratory tract, I decided to go straight to the source and ask my friend Jessica, who happens to be first author on those papers. It turns out to be through the nasolacrimal duct, which is a little passageway that carries tears from the lacrimal tear sac into the nasal cavity. This also happens to be the reason why one sometimes needs to blow one's nose when crying. So Kathy was correct. Jessica also just wrote a great review article on this topic out in MMBR this month, which includes anatomical diagrams. And it's a link to that same paper.
Now,
Hope this helps. All the best, Trudy. Yeah.
Kathy, did we say you were wacko, or did you say you were?
I might have said I'm totally wacko.
Oh,
but,
good. I felt badly.
I think I said that. But
I would never.
But
call
then you said
wacky.
something about tears running down on the outside of your face, and I was thinking, does he not ever cry? But I was telling a graduate student that that the guys didn't really pick up on that, and this was a male graduate student. He said, "Oh, I think every male graduate student has cried." Anyway, that was his take on it.
Yeah, and when people cry, the soundtrack to that is sniff, sniff, right?
Right.
Yeah.
Tears down into the nose.
Exactly.
Right.
So this is cool. We got 2 independent emails on this.
Yep.
Trudy, of course, has been on TWiV. You may know that name, and she is cool that she knows the first author on that paper. So thank you. My last one is just a quick note from our friend Robin, who sends us a Wikipedia post on chicken gun.
Right.
It's not the virus.
No.
But it's a gun that you use to test windshields in airplanes.
Yep.
Chicken gun.
And that's a reference to the TWiV last week with Terry Dermody when you were talking about chikungunya virus.
Oh, that's right. That's right.
They should probably fire them into the engines also. Because out of our window, we could have seen that incident happen, Vince.
Right.
Yeah, there's an old story in the aviation community, a possibly apocryphal story, that when this thing was first developed, the chicken gun, and they were testing windshields with it, I guess the story is somebody like Boeing was using it, and some other company, maybe Airbus, wanted to use the same technique, so they described it to them and sent them some ideas for using it, and a couple of weeks later, the guys at Airbus contacted the Boeing team again. They said, well, this thing really doesn't seem realistic. It always goes straight through the windshield, no matter how reinforced it is. And the guys at Boeing suggested thawing the chickens first.
Yeah, that's great. Nice. Oh, that's funny. Thank you, Robin. I think we should mention this rally for medical research, which is happening on Monday in Washington, D.C.
Yes.
This is, to try and rally to support maintaining the funding for research and not cutting it as part of the sequestration. I mean, I have been told that my grant, which renews May 1, will be cut 10% across the board, which is very hard since I only have one, and it's going to be very difficult to do work, man. So it's not about keeping jobs; it's about doing this work. So
that's
check
okay,
it out.
Vince. I'll work for free from now on.
Okay. I have to cut your salary.
I must tell you, I've always worked for free, so it doesn't really can cut me minus numbers. How's that?
You've always worked for free.
Always.
You had a salary at Columbia.
I did at one time.
All right, a couple of stories I wanted to talk about. The first one, while I was in Manchester last week, I noticed this story, and that is the EMBL, the European Molecular Biology Laboratory, had sequenced the genome of HeLa cells. And so that was on March 11th. Now, as everyone may know, we have talked about HeLa cells a lot here on TWiV. They're commonly used cell line in virology as well as other areas. We use them all the time here in my lab. We grow huge quantities of them. They were of course made from Henrietta Lacks in 1951, a woman who had cervical cancer and died of it shortly afterwards. And HeLa's turned out to be the first immortal human cell line, and they grew and grew and grew. And for years, her family didn't know about them. 24 years later, they found out. They kind of freaked out. And, of course, The Immortal Life of Henrietta Lacks by Rebecca Skloot documents this whole story. So EMBL thought it would be cool to sequence the genome, and they wanted to release it, and they did release the sequence. And everybody freaked out, or many people freaked out. And so they withdrew the sequence because, of course, someone's sequence is their private information, and her family still lives. So I thought we could chat about this and see what everyone thinks.
Mm-hmm. Well, let me ask you this. How do you withdraw a sequence after it's been published?
Take it out of the database.
Yeah.
But people have copied it already.
Well, so it's a lot of bases, so, I mean...
Maybe.
I wouldn't have just downloaded it, even though it's not a lot. I mean, I'm sure some people have, right? But the point is they withdrew it.
Yeah, I think I remember seeing that there had been 15 downloads before they withdrew it.
And are those people obliged to destroy the vial of virus that contains that sequence, so to speak?
I don't know. It's a good question.
Because it's out in the public.
But,
Yeah.
I guess the MBL is talking with the family to try and
Right.
see if they can get permission. I'm not sure the family will be so amenable.
Not at this point.
Not at this point, right?
They'll be just meanable.
So what do you think, Dickson? Should they have gotten permission to publish it?
I mean, given the fact that it's received so much notoriety through the book, and everyone is aware of the fact that their proprietariness is about somebody's own biology, I think the answer is yes. They should have shown some sensitivity, and they should have told them that they were planning on doing this and found out what they wanted to do. And they didn't do it. And so, therefore, they're culpable.
Yeah.
Yeah.
What do you think, Kathy?
Oh, absolutely. They should have gotten together with the family first. And one of the early news releases, according to Rebecca Skloot's article, says that EMBL said, "We cannot infer anything about Henrietta Lacks' genome or of her descendants from the data generated in this study." But then a few scientists decided to prove that that was not true. One took the genome to a public website and translated genetic information and produced a full report of personal information about Henrietta Lacks and her family, which he kept confidential but shared with Rebecca Skloot. So, and we've talked about this kind of thing before, that, anonymizing data is not sufficient. And yeah, at the very least, they should have, contact the family first.
Absolutely.
Yeah, Rebecca Skloot wrote an article, an opinion in the New York Times, and that really sort of made it an issue then. A lot of people saw that.
Yeah.
And she said, the family was not consulted. What do you think, Alan?
I don't know what EMBL was thinking here. They totally screwed the pooch. I mean, this is a cell line. 20 years ago, you could have said, well, we don't know exactly whose cells they are, because nobody had really tracked down the family, and that was an error at that time. But nowadays, there's a freaking best-selling book about the biography of the person who donated these cells.
Exactly.
You cannot possibly plead ignorance on this.
Nope.
And then in the press release, they even say that you can't infer anything about Henrietta Lacks about it. Not only disclosing they know who she is, but also kind of trying to cover the fact that they're maybe not disclosing confidential information. Right. So it seems to me they sort of kind of knew that this wasn't a great idea.
Sure.
And it wasn't hard to find the people who would have to give consent. As I say, there's a best-selling book. You can get in touch with these people. Rebecca Skloot is on Twitter. You don't have a difficult time tracking these folks down. And all you have to do is ask.
Yeah.
You could have saved yourself sequencing the whole genome of this thing. And it would have gone over much, much better, I'm sure, with the family. I don't know whether they would have said yes or no. That's up to them. But I'm sure if they'd been approached in advance and all of this laid out and said, we think this would be useful and we would need your consent, that would certainly have sat a lot better. I mean, putting myself in their position, I'd be much more amenable to say yes under those circumstances than I would be at this point.
Right. And I can't remember exactly where the point was made that somebody said, or maybe it was one in the comment on your blog, Vincent, where somebody said, you could have taken all these pieces that were previously published from the HeLa genome and put them together. So why is it so bad that EMBL did it this way? And it to me, it just kind of strikes me as almost a two wrongs don't make a right kind of thing. Or
that's right.
We now know probably what should be done at the very minimum, and that would have been to contact the family.
Yeah, and also if it were truly possible to reconstruct the genome just through database searches, why bother to sequence it?
Right.
So I don't think that's true. I don't think you could reconstruct the same information
Probably not.
Just from what's already out there. I think EMBL really was contributing something new. This would actually be useful information.
Yeah.
I think science is poorer for not having this information, and I am afraid that we may now never have access to it because the family may quite appropriately say, screw you.
Yeah, I'm sure.
It also, to me, brings up a bigger question for anybody who's going to have their genome sequenced and publish it. So say Jim Watson or whoever.
Or Rich Condit.
Or Rich Condit, exactly. All kinds of relatives of yours might not like the idea that information is going to be public. And so that's an ethical question that, is beyond me to figure out how it's going to be sorted out in the long run. But
I don't know how you do that, because you could argue that if I publish my sequence, maybe in 10 generations, someone else in my family will object. But how can I take care of that, right? I mean, we just... Right. I don't know how you're going to deal with that at all.
That's what I mean.
These issues... These are issues that come up in more than just genome sequencing. Gene therapy.
Right.
Are you giving informed consent for all of your descendants for a gene therapy that could modify your germline?
Yeah.
Well, it also raises interesting questions in the insurance industry.
Yes.
Yeah. What do you think? What do you think, Rich?
I think don't even know what you're talking about.
Hello,
Henrietta's genome. I get it. Okay. Geez, you guys haven't gotten anywhere. We're just on topic number 1 here.
How you doing?
Okay.
Well, it's actually about topic number 4. But
yeah, we did some follow-ups. Yeah.
You have to listen to the whole show, Rich.
Have we already determined that Kathy is not wacky?
Yes.
Or is that later on?
We have.
Okay,
yeah,
good.
We did. Yeah.
Excellent.
Hey, did you want to discuss that?
No, I thought that was wonderful. And I liked her little quip about tears and stuff. Did we get that in?
Yeah.
Yep.
Okay, good. All right.
We're just talking about whether the HeLa genome should have been published, and we pretty much agree that it shouldn't have been.
Yeah, they blew it.
Yeah.
They did.
Geez, did those guys not read the book?
I guess not.
Right.
There are quite a few interesting comments on my blog post, and one of them kind of says, where is it? I find it highly bewildering when scientists all over the world, including Professor Racaniello, use HeLa cells in their labs but start to play moralizers when the sequence gets published. Well, that's not really the point, right?
Right.
A wrong was done, but we don't want to compound it.
Yeah, I mean, you can argue whether a wrong was done.
Yeah, because by the rules
by
of the
ethical
time.
Standards of the 1950s, she was actually treated perfectly fine.
Yeah.
But then you can raise the question: well, is it okay to continue using material that was obtained under those standards? That I think is a whole other ball of wax.
Yeah.
This question of the genome sequence, because the family doesn't really have a claim to the cells of a tumor that would have been thrown in the trash from an ancestor. The family has a very legitimate claim on the genome sequence because they inherited a lot of that.
Right.
And not cool to throw that into the public domain and not even warn anybody.
Yeah, I don't know. As you said, I don't know what EMBL was thinking. I have a feeling they thought this would be cool to do. The cells are out there, but they really should have thought.
I think they got caught up in how interesting the science was and that angle of it. And as I said, this is something that would actually be useful information to have. And unfortunately, I think Embl may have just blown it.
Yeah. Okay. The next story I wanted to mention is this. I picked up on this because we had just been in a BSL-4, and this is the story that this Galveston BSL-4 lost a vial of Guanarito virus.
Oops.
And I went to the page, and that's where the video started playing, Alan. They have a very nice video, and they show people working in a BSL-4, although not us. Right. And showing vials of virus. So, of course, this Guanarito is a hemorrhagic fever virus from Venezuela. I think it exists in rats, but it occasionally jumps into people and can kill them. So they had the virus in the Galveston National Laboratory, which, by the way, we were offered to visit a long time ago. And it's missing. And I thought in light of what we found out in the BSL-4, it would be cool to talk about this, because I think there was a segment of our visit where Ron Corley was talking about how, all the virus vials are inventoried, and the CDC can come in anytime and look at your inventory. If you take a vial out, you have to sign it out and then put it back. So I was surprised that this happened, but I'm glad they reported it.
Is it Guanarito is a urine transferred virus, or is it an arbovirus?
It is not an arbovirus.
Not an arbo.
Not
Okay.
no.
It establishes basically a persistent infection in rodents. It's in equilibrium with the rodents. It doesn't do much to them.
Yeah.
And it's probably urine and feces, basically aerosolized by the rodents, kind of the same way as hantavirus.
Or Lassa.
And if humans have, or it's basically same family as Lassa.
Got it.
So if humans bump up against this in a high enough concentration, they can get infected and get a really nasty disease.
And we don't have any Guanarito virus in the United States.
No, it's in Galveston.
Oh, we actually do now. We just don't know where it is.
Well, so they think it might have been thrown out, and of course the trash is all autoclaved and burned, so
Right.
it's not an issue. Yeah.
So one of the possibilities, and I can't remember whether it was in that TV news report or when my fellow virology class professor presented this as virus in the news, the idea was that, when you take stuff out from the freezer, sometimes, more than one vial sticks to your glove or whatever, and she thought, well, why wouldn't you hear it dropping? And I said, if you watch that BSL-4 video, you know why they wouldn't hear it dropping.
You would not hear an anvil dropping when you're in those suits.
Yeah, we couldn't talk to each other. The yell.
So that was one, hypothesis of how a vial could have gone missing and then, gotten swept up and destroyed or something like that.
Sure.
Or it could have just been a bookkeeping error, as one of the things said. Maybe they'll figure it out, but they self-reported it, and
Yeah.
It's almost like, now I don't know if these sort of procedures are in place, but it's almost like you have to have, in order to, if it's a bookkeeping error or something like that, it's almost as if you have to have two people doing everything.
Yeah.
Okay, so that it's a check on that kind of screw up.
Mm-hmm.
But you guys remember, when you're in those suits, right, you can easily imagine picking up by accident 2 vials and 1 drops, and you wouldn't know it.
Nope.
Because it's so noisy, you don't have any good peripheral vision.
There's
right?
no video surveillance?
Oh, there's video
There
surveillance
is.
everywhere.
There is.
So you could check that.
That's a
at
good
least.
point.
You could, if you could see a vial hitting the floor or what have you.
Yeah.
Well, presumably the freezers have a camera on them, so
see.
maybe they could. I mean, on the other hand, maybe someone took it out to work with it, and then I don't know. So they don't think it's any nefarious thing here going on. It's just a mistake. And
Well, the record keeping in a facility like that, even in BSL-3 or with category with high containment viruses,
Select
even
agents,
at BSL-3 is really a nightmare. But you got to do it.
Yeah. So if you don't know what it's like in a BSL-4, check out our visit. It's pretty cool because no one's ever done that, and you can. There's a part where we talk about inventory of viruses. So
and this is going to be as we've got these BSL-4s around, and they are expanding the number of these labs around. We're going to have stuff like this happen.
Yep.
To me, the thing that matters most is the transparency. Mm-hmm. So they came forward immediately. They went to the proper authorities and they went through the whole procedure that they're supposed to do, as far as I can tell, and then announced that this had happened and notified the public. So, by the way, if you're anywhere near Galveston and you see a vial lying on the ground, you should probably turn it in. But I don't think this is anything. I don't think anybody stole it. It would probably be about the world's worst biological weapon anyway.
Yeah. This is not a good one. I'm not sure there are very many good viral biological
No, there
weapons
really aren't. But
this is not a big deal.
This would be a particularly poor choice.
There's always going to be an error, but it's pretty rare. So
Yeah.
I just don't want everyone to think that these things happen all the time, because look at the movie. There's just a lot of safety practices in place. There are all kinds of protocols for doing everything.
That's not to say that nothing serious could happen or that nobody could steal anything.
Sure.
People can, if somebody were motivated, they could get around various safeguards, but this just doesn't seem like that type of situation.
Right.
And the reporting it promptly is a crucial part of not only the legal requirements but also community relations.
Right. All right, and then we have next a story which maybe everyone wants to hear about, and that is this new human infection with avian H7N9 influenza virus in
But it
China.
was supposed to be H5N1.
Maybe they misspelled it.
That's what we were all worried about.
Yeah, really. I thought the same when H1N1 emerged in 2009. Everyone is waiting for H5N1. So I guess this began in March, but so far 14 people in China, in Shanghai and around Shanghai, I believe, have been infected, and 5 people have died. Influenza H7N9, and this is an avian virus. It's found in wild birds, and it can be found in poultry as well. And it's not really clear how these people were infected. And there's, as far as I can tell, there's no evidence that there is person-to-person transmission. Now, recently, they found a very similar virus in a pigeon at a marketplace in Shanghai. So I guess in Shanghai, they have these open meat poultry markets still, right?
Everywhere.
Everywhere in China?
Everywhere in China.
Yeah.
Sure.
Actually, you walk
Southeast
into
Asia.
when you walk into Walmart in China, the food section has live turtles in tanks and just all sorts of stuff you never see here.
All the outdoor eateries in Southeast Asia that my wife and I traveled through had chickens running between your legs, in fact, right in where you're eating. So you were in pretty close contact with wildlife.
Well, I found
wildlife,
this photograph of a motorbike with all these different birds, dead birds, hooked onto it.
By the way, in Key West, there are also chickens running around.
Is that true?
Hey, they used to have open chicken markets up here in the Bronx.
No, they have them
in
still.
Arthur Avenue. They got rid of this one place. You could walk in, and there was stacked cages with live chickens, and you could pick yours out, and boom, take it home.
There are a number of markets in Harlem that still have that. Yep,
yeah.
Sure.
So maybe this is where these people got the virus, but we don't know for sure. Certainly a pigeon had it, which may have acquired it from a bird or vice versa.
There's one mention in this latest New York Times article that although there hasn't been a confirmed case of transmission between humans, the state media reported today that Shanghai officials had put a person in quarantine who had flu-like symptoms who had contact with a victim.
Yeah.
But the other articles mention couple hundred other people who've had contact. And then, if it's a person who had contact with a victim, well, maybe they were exposed to the virus directly in the same way that the victim was.
Correct.
So that's harder to sort out also.
Yeah.
Yep.
So what are the data at this point? How many confirmed cases and how many deaths?
15 confirmed cases, and 14 and 5 deaths. Everyone.
6.
6 deaths now?
6, yeah.
Where'd you get that from?
That's the Times article, I think, or something.
Because ProMed Mail last night had...
The first line in the Times article, and I saw it on another Google hit.
Yeah, confirmation the 6th person has died.
Oh, yeah, there it is in the ProMed. All right, I got to fix the blog post.
But
this
like
is...
every hour or two,
it's
you're going to
change,
have to make changes.
Yeah.
As of.
Well, the thing
so
is
this is not classified as a highly pathogenic strain formally. Is that correct?
That's correct.
The highly pathogenic strains are those that have the the amino acid in the cleavage site that gives you cleavage and dissemination in the animal.
Most
correct?
of the time, but there are some with that site that are not high pathogenic. So
okay, so the pathogenesis is characterized in birds, right? That's how we classify
so here,
it.
From Field's Virology, low pathogenic viruses cause mild respiratory disease, depression, and decrease in egg production in chickens. And
depression? How do you know if a chicken is depressed?
It's feeling down.
Alan, that's horrible. Those are geese.
Now, high pathogenicity is if it's lethal for 6, 7, or 8 week old, 4 to 8 week susceptible. If it is lethal for 4 to 8 week old susceptible chickens within 10 days following intravenous inoculation with a certain amount of virus. So it's done it's a laboratory test to determine whether it's high or low pathogenicity.
Right.
But this one is low. It doesn't have the multi-basic cleavage site.
Right.
So the HA has to be cleaved in order for the virus to be able to infect cells. and the cleavage for seasonal flu, low pathogenicity, happens in the respiratory tract. But when you put this multi-basic sequence in at the HA cleavage site, then it can be cleaved by proteases in many organs and can be pantropic and more virulent.
So where I'm going with this is that one of the big scary things about H5N1 is that it's classified as a highly pathogenic strain, right?
Yeah.
And then in terms of deaths as a fraction of hospitalizations, it looks pretty nasty because it's like 50 or 60%, right? This is the H7 strains on the surface maybe don't look as exciting because they're low pathogenic strains, but right now at this point we're talking about, of the hospitalizations, confirmed cases, 40% deaths. So the pathogen, what I'm wondering is whether the higher low pathogenicity that's characterized in birds really has anything to do with what we see in people.
Well, and to throw another issue into this is not just an H7N9. It appears to have the guts of an H9N2, right?
Right, that's correct. It's a reassortant. So
it's a reassortant. It's got H7N9 on the outside, H9N2 on the inside.
Yeah.
Essentially.
So Brian Kimball, who actually works on H9N2 at the University of Maryland, he put on Google Plus that he looked at the sequence and it's a reassortant. So I asked whether all H7N9 viruses, so these you get from birds typically, right? Are they all reassortants or are they not? And I don't know if we know. Brian said we're not sure, and it's not clear. So I'm wondering if this was a chance reassortant, and that reassortment made it able to infect people, or whether all viruses that infect birds, or many of them, also reassort.
So I have a question, and it relates to the H5N1.
I'm sorry, before you do that.
I'm sorry? Okay.
I got an answer. Yes, so
Right.
GSGS over on my blog said there are, from birds you can get reassortants, but it's not clear how they're related to this one.
Right, so it looks like H9N2 shanghaied the capsid of H7N9, right?
But
Good title.
not bad. So going back to the H5N1, remember the epidemic, or if you could call it that, was particularly fatal for little kids. So the age group was skewed towards little kids that would probably see
What epidemic are you talking about?
the H5N1 deaths from bird flu occurred mostly in small children.
Are you sure about that?
Yeah.
But there are also old people getting killed by it.
Well, but
it was more of a W-shaped curve, kind of like the 1918.
That's right, exactly right. I agree with Kathy.
You say young, middle, and old. Is that what you're saying?
Yes.
Yeah.
Young was typical, very young children. Elderly was typical, and then there was atypical peak in the middle age range.
Well, we're only talking about 600 cases now.
Exactly.
Correct.
Well, that's right. So, but the point I'm raising is that what does the epidemiology look like in this particular case for the H7N9?
There are only 14.
Yeah, well, of those people.
Well, I don't know. I didn't go through all the deaths. Has anyone?
I don't think it's worth doing at this point. The one's a 4-year-old, one's an 84-year-old.
Keep going.
Yeah. N is too small.
Yeah, well, what about the 17 or 16 or 14 that are
It's too early.
sick?
But H5N1 attacks are lethal in pre-adults and young adults, while older victims tend to have mild attacks. Okay? So it's not really a W, but it's okay. Yeah, I think it's too soon, Dickson, to know here.
Well, I just thought you could
But I'm not saying you shouldn't have asked. No.
No, good question.
It's fine. But
I'll save it for later.
We just like to answer. So now a couple of other things. They are killing lots of birds in Shanghai. They're culling because if in fact these infections came from birds, which seems likely, they should cull to stop the transmission.
Yeah.
Another thing, I asked Peter Palese what he thought about it, and he said it doesn't have the multi-basic cleavage site, and he said also it does not have a serine at position 61. As does the 1918 virus. And he said this is a marker for human virulence in some animal viruses. Brian Kimball also said that it has a amino acid change, which would in the HA, which would suggest that it can bind human sialic acids. That is the alpha 2,6 sialic acids. So the avian strains prefer alpha 2,3, and so the fact that the avian strains in nature, some of them are trending towards liking alpha 2,6 sialic acids, and these H9. Which ones did they say are trending towards alpha 2,6? So this one is clearly there as well. So that means it can more readily infect the upper tract, which has in humans, which has alpha 2,6, as we can see from our nice picture from that paper that we talked about before. Alpha 2,6 is in the upper tract all the way down to the lower tract. So that would
that slightly increases the odds of being able to transmit person to person.
Yeah.
I have to correct myself about the W curve. I was thinking, I'm pretty sure, of the 2009 H1N1.
Yep.
Right.
So that's where I got that. Sorry.
Yeah.
As soon as you said 600 cases, I thought, oh wait,
right.
wrong virus. It's easy to see why our students got confused on the exam.
Right.
All those different viruses that we threw at them.
Indeed.
See, there was an H in there and an N, and then a couple of
That's
numbers.
That's right. Some numbers.
Yeah, it's definitely, I was thinking of the H1N1. I have my lecture notes up now.
I think the little kids were particularly susceptible to the lethality of H5N1.
So there's like 16 HA subtypes out there in birds, and 9 neuraminidase subtypes.
Right.
And nature has been doing this experiment, trying to infect humans
Shuffling the
for
deck.
probably thousands of years.
Yeah, I don't know about that.
No?
Why is it nature trying to infect humans? Just trying to infect anything that'll let it replicate.
Yeah, anything.
Yeah.
Sure. But only HA1 through 3 and neuraminidase, there are 2 or 3 subtypes that have shown up in people.
2, N1 and N2.
N1 and N2. So just 3 of the HA.
That is bona fide human influenza viruses that
established in humans,
Yeah.
transmissible in humans, get a foothold in humans.
Got it.
So we're being exposed to these things probably all the time. Okay, and my guess is that part of what's going on here is we have better reporting, better diagnostic tools, so we're seeing more evidence of these other subtypes occasionally, infecting humans, but not really getting anywhere. And my guess is that this has been going on.
Could be.
For millennia, perhaps.
Yeah, but we haven't
and
monoculture.
only 1 through 3, HA1 through 3, and N1 and 2 have actually established a foothold. So what is it? Is it those virus surface proteins that really are limiting the entry into the human population, or is there something else going on?
Or have these just been unlucky?
Well, the other thing
a lot
is
of things.
Modern life is much different than life before we started to have agriculture. And so having all these chickens all in one place or ducks all in one place is a modern example of human life. And we weren't like that 10,000 years ago or even
Right, but we were
5,000.
like that. We were like that 500 years ago.
Sure. But there weren't as many people.
Right, but if you go, we're talking about these open air markets in China.
No, that's true.
Those are not a creation of the past decade. Those have been around for a very long time. And I think Rich's point is valid, that there's been a lot of opportunity for all of these HA and neuraminidase subtypes to interact with humans, and of course they're reassorting all the time because it's flu, and yet somehow only a small subset of them has successfully jumped into humans.
Yeah, but
is that luck, or is that something about the virus's biology?
The world's greatest vector, the 747, wasn't around for that long. So this virus had no way of getting out.
The viruses evolved, and they just test new species, and whatever works for that moment continues.
Yeah.
Sure. That's all.
Actually, the other thing that I don't know is sort of what the prevalence of these different subtypes is in the environments that are really bumping up frequently against humans. So in poultry markets and that kind of stuff, how often do you have subtypes other than 1 through 3 and et cetera
Right.
very, very common. I don't know. I mean, yes, there's 16 subtypes, but it could be that most of them are in ducks and stuff that wild ducks that we don't necessarily run into much. I don't know the answer to any of that.
Right, and there's something up with agriculture in China at the moment. You probably saw the story about the pigs in the rivers.
Yep.
Yeah.
That is apparently not normal. And
so let's remind people, those are the pigs outside of Shanghai who died of something.
Circovirus?
The farmers threw them in the river and they made it to Shanghai.
Right. So there were hundreds of pig carcasses washing down the river.
18,000.
Thousands. Sorry.
18,000.
Yeah. So this is a huge mess. And one of the stories that I saw on that suggested that one reason they were being thrown into the river was that the normal dumping sites for pig carcasses in the rural areas were full.
Right.
So there's something up with the, and I don't know. Somebody really ought to do a deep dive on this story and find out what the weather patterns and disease rates and such have been in the rural areas there, because there's something going wrong with the pigs.
They fed them too much. They fed them too much melamine.
Right. I'm not. They did in fact test pig carcasses for the H7N9 and didn't find it. I wouldn't expect a direct connection like that. But if you've got some ecosystem disturbance going on that's causing farmers to lose pigs, maybe that's also causing some other animal husbandry problem and getting new viruses into the birds.
Alan, this sounds like a toxic event. Something in the food or something like this just affected those pigs all at once.
Maybe. Or maybe a disease event, or maybe a weather event.
Didn't they blame it on circovirus infection? But then they withdrew.
Well, they did isolate circo, but we don't know if that was.
We don't know if that caused the deaths.
Yeah, exactly.
So I was just looking at Brian Kimball's blog, and he mentions that pigeons do not typically spread influenza, so he thinks they're swabbing away there, and they'll find it in poultry eventually.
Schwabbing away?
Schwabbing. He's got a nice table of all the cases so far.
I think they would be squabbing.
I meant to say that. Of course I did,
Over on my blog, GSGS, who has followed us for a long time, posted a couple of things which are worth mentioning. He says he looked in GenBank for sequences. He got 20 wild bird H7N9 sequences out of the 5,000 or so sequences at GenBank. HAs and NAs switch frequently in wild birds, and H9N2 is also in wild birds. So H7N9, HN92 reassortments may well happen in wild birds, but it clearly seems that this new H7N9 reassortment happened in poultry, probably chickens. I don't know why he says that. I guess because there's nothing closely related in the database. And he also says low pathogenicity viruses, 30% ? Or sometimes convert to high pathogenicity after months or years of evolution in chickens. That's why they cull even with low pathogenicity outbreaks like this one. So, Dickson, is this the next pandemic strain?
You're asking the wrong person. Last time I had to guess at this one, I said much ado about nothing, and that was
you were wrong.
Completely wrong. I mean, it swept the entire Earth, so forget me. Leave me out of this discussion.
But that's why we want your prediction.
Come on, say yes or no. I want to just see.
I think no. I think no.
That's fine. That's fine.
All right, everybody, hunker down.
Why? Yeah, right. Because it got. Sorry, I'm very slow.
That's right.
I'm slow. All right.
You're slow.
The last pandemic strain in 2009 also began in April, right?
Yeah.
And we're in April, dude.
That was in Mexico, though.
Rich Condit, what do you think?
No way.
No way?
No, good.
No way.
2 out of 2.
Alan Dove.
I'm going to go with no.
Excellent.
I don't. I mean, I could very well be wrong, of course, but
Yeah.
I'm leaning no.
Kathy Spindler.
I'm going for no.
Yeah. I agree. I think this is not a pandemic strain. I think,
It's certainly worth watching,
absolutely.
figuring out what the ecology
So
of this
we're all
thing
going to get
is
this one
in
wrong,
wild.
that's what you're saying.
That's fine. But the Times article, they say U.S. is starting to work on a vaccine, which is probably okay.
Why not? That's
not
okay.
too bad.
Yeah.
Sure.
Okay.
Just
Sure,
to be
and if
careful.
nothing else, I mean, it's good to practice your vaccine development and have these things sitting around just in case. And this virus could contribute genes, as in all of these cases with H5N1 and other flu viruses. It could contribute genes to the next pandemic.
Right.
The problem is that if it starts to spread, it's too late to start making a vaccine.
Yes.
Yeah. Well, even now, if it's, I mean, the thing is, when the H1N1 emerged in 2009, that spread really quickly. And like by now, it would have already spread. So this is not spreading like that. And and some people say, well, it has to acquire transmissibility. That's not the way this works. You get the virus that's ready to go in people, and bang. I don't think it evolves to that point in people. So that's why I don't.
You get the virus, then you get the transmissibility, then you get the pandemic.
That's right.
So that's why I think this is not it. We would have seen it going. I think these people a lot of them are in contact with chicken. In fact, I just read on Brian's blog that one of the women who died was slaughtering poultry in the market. So that's a good way to get this infection. What I would think would be really interesting would be to look in the genome of these individuals and see if they have some polymorphisms that make them particularly susceptible.
But ask their families first.
Yes. We don't have to publish it.
Yeah.
Dear.
Yeah, I think that would be very interesting.
Mm-hmm.
But I wanted to point out one other thing that I've noticed with this and with the H5N1, and China has gotten vastly better at transparency with things like this.
Here. Mm-hmm.
Yeah.
Compare this to SARS.
Yeah.
And if you let something, I mean, obviously, it appears that the lesson was learned. If you let something like this fester and you don't talk about it to anybody, it could get really, really bad.
By the time it is, then you wish you hadn't.
Yeah, but here, we've got practically real-time updates going to ProMed Mail and sequences deposited in timely manner, and WHO's on board, and it's just much, much better handling of this.
Yes.
Yeah, just thinking about the chronology of it, the first death was March 4th, next one was March 10th, and the third one was March 27th. And, that wouldn't necessarily even be something that you would pick up on.
Yeah.
But, in fact, they did, and they made the report on April 1st. So they seem to be much more on top of it.
These coronavirus EMC is the same thing. You've had, 15 cases so far over a long period of time. I don't think that's going anywhere. It would have taken off already.
Correct. I would. Totally.
Kathy, did you paste in this H1N1?
Yeah, that was the one. Those pictures are from as of October 2009. And so I was just confirming why Dickson and I might have gotten confused. There was this high death rate for the middle age groups from the 2009 H1N1.
Yeah.
So,
the last thing I wanted to mention was, while I was reading Brian's blog, I came across a paper on which he's a first author. It is in PNAS. It's called "Compatibility of H9N2 Avian Influenza Surface Genes and 2009 Pandemic H1N1 Internal Genes for Transmission in the Ferret Model" by Kimball, Sorrell, Shao, Martin, and Perez. And this is a 2011 publication where they basically made reassortants and asked, if you have the internal genes from the pandemic H1N1 and you put the surface from H9N2, does that transmit among ferrets? And the answer is yes, sometimes. Some of the viruses do, but not all of them. And so it just shows that these... of course, we know that ferrets are not humans, and neither are chickens, but it tells you that these are compatible. This was an interesting paper because this fell under the ferret radar.
Yeah. And when was that? 2011.
Yeah.
When were the Kawaoka and
so the brouhaha began in September of 2011.
July or September, yeah. Well, we found other papers that flew under the ferret radar as well.
Yeah.
We discussed one on TWiV.
In fact, I had visited Peter Palese last year, and he was complaining. He said, look at this. Look, he showed me this paper. Here's ferret transmission, and nobody cares. Of course, these are low pathogenicity.
You mean one flew under the ferret's nest?
That was
but I wondered, as I was discussing earlier, low and high pathogenicity are assessed in chickens.
That's
I really wonder what that means in humans. I don't know.
Well, Rich, are chickens closer to humans than ferrets?
You're asking me?
Depends on whether you live on a chicken
Depends
farm
on the
or a ferret farm.
Anyway. All right, so there you go. Anything we missed that we should have talked about?
No.
No, that's it. So we all think it's not the pandemic strain. It's interesting. It's worth studying.
So everybody stock up on canned goods.
Right. Right.
If you cook your chicken, you can eat it. That's not a problem, right?
This is all
of course,
But
if
don't stock up on live chickens.
Yeah.
Let somebody else kill it for you, at least.
Get them frozen. Yeah.
Get them frozen.
All right, let's do some email. Let's begin with Rich Condit. Can you read Bill's?
Sure. Bill writes, "Hi, it is an unseasonably warm 5 degrees C here in Spokane. During the last episode, an individual wrote in to find the best way to learn basic cell biology. I agree that Albert's molecular biology of the cell or Lodish's cell and molecular biology are far too advanced for most beginners to biology. However, books like Albert's, and these are all et al.'s, Albert et al., Lodish et al., Albert's et al.'s Essential Cell Biology should provide the basic fundamentals of biochemistry, central dogma, cell anatomy, and cell function to get a beginner on track. An older edition can be picked up for a tenth to a 20th of the price of a new edition and is still perfectly adequate for their needs. Great show, Bill." I'm not familiar with that, the Essential Cell Biology.
Sounds like an excerpt from the longer book.
Yeah, might be worth a look.
Yeah, so the listener who had asked, there you go. You can check that out. You can get a used copy cheap. All right. Alan.
Ricardo writes, "Hello, TWiV gang. Something as beautiful as insightful," and he's not referring to us. Pointing to this website, information is beautiful.
This is cool.
Oh, yeah.
This is really beautiful.
Yeah, it's cool graphs, and in many cases they've stripped everything except the graphical elements.
So you can click on them, and then you. Like this one...
20th Century Death looks like this abstract art piece at the top, and you click on it, and it gives you the causes of death.
And you can click more, and you get bigger and bigger. It's very cool. It's a nice way to present data. I like that.
Yep.
It is beautiful.
Yep.
And he wasn't talking about you, Dickson.
No, definitely not.
Kathy.
Are we up to Steve? Viruses in podcast from nature.com. The new coronavirus has pandemic potential. Where did it come from and how is it spread? And so Steve has sent the above link to the main podcast page for this episode. The audio link for the segment on coronavirus is down along the left-hand side of the page at the 1912 mark. I couldn't pull a direct link to the specific coronavirus audio segment, otherwise I would have sent that. So evidently Nature does podcasts, and this is one about the new coronavirus. And then he has another link here for a receptor for the new coronavirus found, which is likely the paper that we covered a couple weeks ago.
Right.
Yes.
Right. Okay.
Thank you for that podcast link.
Yeah. Hey, Dickson, can you read an email?
I can, but I can't get it up on my screen now because I can't get back to my email.
Why not?
I don't know. I'm looking at these beautiful graphs, and I'm wondering how to go backwards.
You work on that. I'll read this one.
You think?
Yeah. This next one's from Ed. Dear TWiV hosts, I'm avoiding repetition this time by not listing names in case Alan gets too much attention.
I never get too much attention.
No, he doesn't.
I think once you had your name twice, right?
Yes.
A couple of papers came out in the March 1st issue of the Journal of Immunology, may help to shed some light on sensing foreign DNA in the cytoplasm. One is a review by Atlanand and Fitzgerald. The second is a paper that purports herpes simplex virus capsids are broken down in the cytoplasm of myeloid cells, e.g. macrophages, by the proteasome, whereupon DNA is released to the cytosol for detection by IFI16. Provides a link to that, too. I guess, Kathy, these are both the same link, so maybe I should contact Ed. Or you think you found the paper that is the right one?
Yes, but it's not in Journal of Immunology. The second one is in a different journal, so you might want to check with him.
I will ask him. I think we're going to... And then there was last time the paper by Christea at Princeton. I think we should do those all together at some point. Talk about sensing, because clearly everyone heard me say that you have to find a nuclear sensor, and now people are.
Yeah.
Isn't that cool how they do the research that we ask them to do?
Yes.
They've already done it. That's all.
I'm just kidding. Dickson has left the room.
Okay.
Is he still looking for his email?
I don't know what he's doing.
It went down the hall.
It's gone. So we'll just continue without him. Have I read one? I don't remember. Did I just read that one?
I think
I
you just
did.
read that one.
So we're back to Rich.
Cool. Lucky Fractal writes, "Hello, Dr. Racaniello. I'm assuming you've probably read every virus-related book out there. Even so, I felt I had to send you this excerpt from, quote, Virus X, Understanding the Real Threat of the New Pandemic Plagues, unquote, by Frank Ryan. It struck me as a very poetic description of viruses. Feel free to read it in the podcast or not." And here's the quote. You want me to go ahead with this?
Sure. It's kind of neat.
"Viruses are also uniquely vulnerable. They cannot come to life other than within cells of their destined host. Consider the uncertainty this creates. To survive, a virus must constantly adapt and evolve. Every act of infection involves a battle for access, a means of circumventing the phagocytic defense system, followed by the adaptation to the adamantine set play rules of the genomic landscape itself. Once established, it has to compel the reluctant genetic mechanism to adopt the progenitive program of its own genome, then outwit the attempts of a now responding and extremely resourceful cellular and humoral immune system in order to survive once outside the amplifying cells. It had to fight off a myriad such forces bent on its destruction while discovering a way of leaving this host and finding another. One solution to this, perhaps a very common solution, is for the virus to discover a way of cohabiting with the infected host for a very long time, perhaps for the remainder of the host's life, with its implications of de facto eternity through an evolution of vertical transmission from parent to offspring. The inference of such a relationship between two such dissimilar forms of life is a much greater intimacy of give and take than the anachronistic notion of pure predatorial aggression. Such refinements of evolutionary partnership must flourish in the fields and jungles, in the oceans, the soil, and the air, where over eons of evolution, viruses have explored a multitude of wonderful, if often mysterious, genomic archipelagos, establishing relationships with every species, extant or extinct, that has ever evolved on Earth."
All right, look. I don't think it says anything, and I don't think most people are going to understand it. I think it's way too complicated. What the hell is adamantine?
Hard. Adamantine is it's an
antiviral.
Might be an
Yeah, that's what
alchemist's term for diamond or something hard or something.
Alan, what do you think of this writing?
Adamantine. It's very flowery. I mean, I get what he's getting at. I think he uses too many words to get to it.
Yeah, I agree.
You know, but the concept is that the virus is not. It's not just a predator. It's not just coming in and eating the cell. It actually has to, because it's completely dependent on the cell for its replication, it has to reach an agreement.
Yeah.
In a sense, it has to figure out what it can do and what it can't do, and there's this negotiation going on evolutionarily. But I'm not sure that the language he's using really helps get that point across.
I think a lot of people. I think with science you have to be a little less flowery because the science itself is hard enough.
So what's the maxim? Don't use a $10 word when a 2-bit word will do.
Yes,
that's right.
So this week in vocabulary, adamantine is the first pronunciation, although adamantine is also acceptable, means unbreakable.
Okay.
And it also says literary.
The drug is amantadine. I forgot.
Yes,
it doesn't have a D there.
From a fungus.
I haven't read this book. Has anyone? Probably
No.
not. I might look at it, but I think Lewis Thomas used to write about this, this negotiation between host and pathogen, and he wrote beautifully, very simply.
This is true.
And a long time ago as well.
Yeah.
This is all right.
And
also Carl Zimmer.
Yes.
Yeah.
Yes. Carl would not be flowery like this.
No.
So viruses have some problems to deal with, and it is a jungle out there. But I'm not convinced that it's really any more difficult, that there are any more negotiations that a virus has to take on than a bacterium or a human or, a flea or whatever. It's a jungle there for everybody.
Yeah.
Oh, yeah.
Right, but I think this perspective of parasites and hosts having, there's parasitism and there's symbiosis, and there's kind of a gray area in between, and even a pure parasite has to reach some kind of accommodation with its host in order to ensure that the host lives long enough to propagate the parasite.
Right.
It's a worthy concept.
Yes.
All right, let's read one more. Dixon, are you reading?
I can read now. I can read. Yes,
sure,
I can.
Okay. Can you read
Jessica? I can. Jessica. Dear TWiVers, thank you so much for episode 217! I'm a PhD student working on influenza at the Robert Koch Institute in Berlin. I just got back from Berlin, and I absolutely loved this episode. Actually, I listened to it twice to make sure that I got everything. Furthermore, I wanted to ask you something. Did you ever think about interviewing someone from the CDC in Stockholm? No matter if it's about a recent story like the novel coronavirus or infection epidemiology in general, I think it would be an outstanding episode. I had the opportunity to meet Andrea Amond at a conference in Berlin last year, and she is totally great. Listening to her about her work during the SARS outbreak was an absolutely fascinating event. I will fill in there as editorializing. I am sure that she would be a perfect guest for TWiV. Maybe you'd consider asking her, and then she gives her email address. Last but not least, here is a reminder that you, Vincent, are invited to be a guest speaker at the ZIBI Summer Symposium 213, quotes, One Health, June 10th to June 11th, 2013, here in Berlin. It would mean a lot to me and all TWiV listeners in Germany if you could make it to Berlin. Please find attached the official invitation letter. All the best from Germany, and many thanks for your wonderful podcast that makes my long hours of marathon training in the park so much more interesting.
Wow, marathon. Huh?
Jessica.
I'm sorry, Jessica. I have to go to study section on June 10th and 11th, along with Rich Condit in Seattle.
Well, maybe Alan and Kathy and I could go.
Oh, I'm sorry. I wish I could, but you and I are going to be in Seattle, Rich.
Well, I know I'm going to be there. I guess we're going to be. Yeah, we have to figure out a TWiV.
Yeah, we'll do a TWiV from Seattle. Sleepless in Seattle is the name of it, right? Is that a movie?
It was.
I didn't see it.
Yeah. Oh, it's a wonderful.
Oh, you didn't see it? Oh, you need to see that.
It's quite nice.
Really?
It's very touching.
Yep.
There's so much to do, right, Vincent?
Yeah. I get itchy when I sit down to watch a movie because I just start thinking of all the other crap I have to do, but I can't pay attention.
Maybe on the way out to Seattle you can watch it.
I'll probably be reading papers for the TWiV that we're going to do there. Let's do some picks of the week. Dixon, I know you don't have one, but think of one. You've got a few minutes. We're going to start with Kathy.
Think of a pick.
What do you have, Kathy?
So I picked some YouTube videos that you can get to from the NIH NIAID site. And when I first saw this, it was very soon after the one that Alan picked about drug development. And they've got one about drug development with a slightly different take on it. And the reason I was on the NIAID website was I was trying to figure out, this was almost a month ago, how the sequester was going to affect everyone's grants, and all they could say was, check your own institute, and NIAID didn't have anything, but these videos caught my eye. So they have some about how pandemics occur. They have one about Ebola. They're just very short videos that I think people might like to take a look at.
Cool.
I looked at these, and I think that they're terrific. I looked at two of them. I looked at one on, I forget one of them, but the other one was the setting up a camp in the Congo,
right,
to look for reservoirs for Ebola. It was great. They're very nice videos. They're terrific.
Cool. Rich, what do you have?
This is in the category of the crossover between science and spirituality again, one of my favorite topics. This is a story, a short basically audio blog that I heard on NPR titled How to See the World in a Grain of Sand by Adam Frank. Adam Frank is an astrophysicist at the University of Rochester who is also a writer, and apparently he's blogging for NPR and is going to have a number of these audio bits coming up. And he starts off by quoting one of William Blake's poems from a couple of hundred years ago, to see a world in the grain of sand and a heaven in a wildflower, hold infinity in the palm of your hand and eternity in an hour. And he says, yeah, right. Can we do that? And his answer is, yes, I can. And the reason I can is because I'm a scientist. Right?
Nice. Yep.
And then apparently he's going to go on in these future stories, and he's going to start off with seeing the world in a grain of sand. But what I really like about this is that he has even a more, in this particular piece, even a more essential definition of science than I've considered before, and that is, if for him at least in this piece, science is just noticing things.
Yeah.
Paying attention.
Yep.
Okay? I really like that. And so, his theme is going to be, using, everyone can be a scientist, really, just by paying close attention to what's really there. It's cool.
Mm-hmm. Yep. Very cool. I like the way you read poetry, Rich.
I do too, actually.
Nice. Very good.
With emotion and passion.
Alan, what do you have?
I have a completely silly pick. Surprise, surprise. I don't know. Was my last pick also a silly one? I'll have to do a serious one next.
Oh, come on.
Anyway, these are insects made out of Lego pieces.
Cool.
Very cool.
And somebody's got
Nice.
a series of these on Flickr.
Wow.
They've done some of them. They've got them mounted in nice cabinets,
Riker mounts?
Riker boxes, like
Right.
a collection.
That's cool.
How funny.
That's very cool.
Very nice.
I have, my kids had lots of Legos. They're all in one box.
Right.
It's terrible. Maybe someone should make these things out of it.
Yeah.
Dixon, did you come up with something?
I did, actually, because I was very impressed with the fact that a city can devote itself to science. And I was privileged to be asked to speak at the Edinburgh Science Festival.
Edinburgh,
however you want to pronounce that.
That's not me. They told me. That's how.
Edinburgh.
Yes.
Edinburgh. Okay, Edinburgh is in its 25th year of Science Festival. So I highly recommend going to their website and checking out the topics that were discussed throughout the festival. Quite impressive. There was one called Walking with Scientists. I don't know if you've ever seen Walking with Dinosaurs, but this was essentially a science tour of the city to give a take on things that you take for granted, but do you know where that water comes from? Do you know what the air is purified over here? That sort of thing. So I was just taken by the whole idea of an entire city devoting itself like that.
Nice.
Yeah.
Did you like Edinburgh?
I was colder than you know what. I mean,
Well, I was there at the same. I was
you
in
were.
Manchester at the same time.
It was
as you were there.
ridiculous,
Yeah,
and
it's quite
I
cool.
didn't get out much. But I did happen to just casually walk past the Elephant Cafe. Does anybody know the significance of that?
No.
It's where Harry Potter was first conceived.
Ah.
Really?
Yeah.
What? She had the idea there. She was
That's
sitting in
right.
the cafe?
That's right. And it's got a big sign on the bottom where Harry Potter novels were first thought
That's
about.
not good. Thank you, Dixon. My
welcome.
pick is a blog post by Cassandra Williard, who is a science writer, and she's here in Brooklyn, and I've met her and spoken with her. And she had sent me yesterday a question by email. She had gone to a talk by an anti-vaccine person, and she had sent me what he said and wanted me to comment on it. And so this is her blog post, Vaccines, Viruses, and the Anti-Vax Movement. And just parenthetically, what a great name, Cassandra Williard. So she has to be a writer. So this fellow, basically, his argument was, our genomes are almost half viral, therefore they protect us and we don't need to use vaccines as a consequence. And she said he basically said this to the audience and then told them they had to make up their own minds. And she said there were lots of pregnant women in the audience. She felt really bad. So her post is basically saying how terrible this is and how misleading he was. She says, I'm always in favor of a critical analysis of the evidence, but that's not what this fellow gave me. He seemed to cherry pick, misunderstand, and misinterpret. He wove science together with anecdote and opinion. In a nation where just 28% of adults are science literate, the combination can be deadly. So my quote was, Pelevsky either does not understand virology or he is bending the truth to support his reasons for not immunizing. I have never seen such a flagrant display of ignorance with respect to vaccine arguments. There you go. So I think we ought to look at this, because if anyone went to this, you should know that it was wrong. Please. This is terrible. It's really, really bad.
This type of thing is not... I mean, she does a nice post and discusses this experience of going to this thing, and it's obviously an absurd argument that they're making. But these types of groups are really more like a religion than anything else. You're not going to reason them out of this.
So you guys all suffered through the presidential debates. I did too, and some of them I watched, and some of them I didn't watch. But what would be wrong with getting some neutral body like National Public Radio or Public Television to set up a debate of, six people. Here are the virologists and the vaccinologists on one side, and here are all these kooks on the other. I mean, here are all these wonderful people on the other side, and have each one give their point of view and set it up as a real national debate. Because right now, the country is teetering on, yes, I should, no, I shouldn't, yes, I should, no, I shouldn't, and they have no good information that's coming right at them, right in their face, from both groups at the same time.
No offense, but I think that's a terrible idea.
Because
if you frame it as a debate, it implies that there is a controversy, which there is not.
Yes, well, but they've done this, Alan. Right. They've done this for creationists versus evolutionary
and
theory.
Look how well that's worked out.
No, but it always...
No. This type of approach, I can tell you, it's absolutely not the right approach, because you would be giving airtime to views that should not be given airtime.
They're already getting airtime.
Right, but don't give them any more.
No, but this would discredit them, I think.
No, it
wouldn't.
You don't think so?
These are people who are well versed. I mean, you're dealing with professional shysters, who are very well versed in confusing the issue just enough that people who are not scientifically literate will go away thinking there's a controversy. Doubt is their product.
There's no virologist out there or vaccinologist that could ever dispel that?
No, because the nature of the response that they're going to give is going to be one that you can answer, but you would have to use terms that the audience will not be able to understand in the allotted time. That's the whole strategy. Seriously, it is an unwinnable debate when you get into it with these professional fraudsters.
Seth Mnookin discusses this in some detail in his book, The Panic Virus, and how frustrating it is to see these sorts of debates that have the effect, really, of lending credibility to the anti-vax movement, where it shouldn't be lent credibility. And quite often they're put up against scientists who, are good scientists, and they won't say anything with certainty, right?
But it doesn't even matter if you pick the scientist with the best public presentation skills. I could not coach somebody for this debate, and that's something I used to do. It's not a winnable debate because the audience is not well versed enough in the issues to be able to tell which one is the fool.
Yeah.
And your opponent would be using the same terminology and would sound credible. And that's the whole problem.
Yeah. Did some of you must have heard about this letter to my anti-vax father? That's been going around on Twitter. That was very good. Did you read that, Alan?
I did. That was that actually. I meant to bookmark that as a possible pick, but maybe we're doing it now.
I don't know. Might go. We can save it for another time. It's fine. We actually have a listener pick that's related to this.
That's relevant. Yes.
Yeah. So this is from Mehul, who says, "Hello, Kathy. I went to a vaccine dinner club hosted monthly at Emory University, in which we invite various outside speakers to discuss research related to vaccine. Today we hosted Diane Griffin from Johns Hopkins University. She gave a wonderful talk about measles virus and innate immunity. Before her talk, though, someone, I did not catch his name, gave a brief presentation about voices for vaccines. a nonprofit organization led by parents to advocate for vaccination. Great cause, and I am sure they would appreciate a TWiV bump. So we will give them a bump.
Yes. I've looked through this website, and it's quite nice.
Mm-hmm.
And this, by the way, is what I think is the correct response to the anti-vaccination nonsense. Mm-hmm. And as a scientist, I look at this and I say, well, these people are just telling anecdotes.
Yeah.
But that's the issue. They're fighting anecdote with anecdote.
Right.
And
one of the lines from the blog from the page is, stories shared between parents are often as compelling, if not more so, than information we receive from research papers, newspapers, even doctors' visits.
Yes.
Yeah,
right.
yeah. I think
that's true.
That's the Alan, you had given us a pick before of the flu.
Yep. Families fighting
yeah,
flus.
Same thing. Yeah. Mm-hmm. And Dickson, are you okay? You seem to be very sad that your idea was debunked.
No, it's not my idea. I'm very sad to realize that you guys may be right.
Yeah.
Right.
And that actually, it's hopeless. It's absolutely hopeless for half of the country.
No, I applaud your desire to have an open, rational discussion about this, but the problem is that we're past the point where rationality is.
Alan, don't you think people can spot shysters when they hear them?
No.
Absolutely not.
No. If they could, we would not have shysters.
No, this is too long a discussion to waste time on TWiV about, but all right.
No, I see where you're going with this.
I know you do. I
right. You see where I'm going with this.
Yes, of course.
All right, one more pick.
We're going to go for a beer.
One more pick from Stephen. As you read 2 FYIs I sent on episode 224, I'll suggest a listener pick of the week, the free Nature iPhone app. Both my suggestions read last week came from the Nature app. I don't subscribe, so I only get the free parts, Nature News and some Nature Communications, but I usually find something interesting each week. And then this is, I think, what he had probably sent us a story about how the Google flu trends didn't work this year.
Right.
So he adds an addendum. The discussion of how to cope with online anti-science fit with the Google flu trends link I sent. On a technology forum, a participant wrote, Google does a better job than the CDC in tracking flu outbreaks. It turns out that an increase in people searching for flu symptoms remedies is more accurate and timelier than the CDC review of hospital reports. I responded that the comment is just wrong and cited the Nature News item you discussed on TWiV 224. I have nothing against Google's interesting experiment, by the way. The reader responded, the article assumed that Google got it wrong, but it's quite possible that Google got it right and the CDC got it wrong. Of course, that could be true, but where would you place your bets?
And also the argument seems to be, let's begin by assuming my argument is correct.
Yeah.
Yeah.
Where CDC is also basing it on virus isolation, right?
Yes.
So
the CDC has never had a problem with accuracy, or much of a problem with accuracy in their flu reporting. It's speed that they're not so great at because of the amount of time it takes to be accurate.
Vincent has the right idea. I think we have to speak up for rationality and keep speaking up. Anti-vaccination sentiment, while important, is merely one tip of the iceberg. Wow, we have a theme here in picks, don't we?
Yes.
Mm-hmm. The last thing I wanted to do, Alan, we had quickly left the Cassandra's thing, and you'd said you'll never win. But I still think she had to write this, right?
Oh, yeah, absolutely. I mean, she went to this event to see what's being said on that side of the discussion, and she saw it and reported it. And yes, absolutely. I think it's a great blog post and very informative. And
I like her last line. I left the lecture dismayed. Seth Mnookin, author of The Panic Virus, tried to make me feel better, but it didn't really work.
Seth is also over there in Brooklyn, so I guess they both went. That's cool. Yeah. Maybe we should move the TWiV studio to Brooklyn.
Well,
that's too far for me.
That's where the secret science club
So this
meets.
was a TWiV where we didn't do a paper. We just had topics. Is that cool? Do we like that?
You did a paper. You did a little bit of a paper.
It was a tiny bit.
That's okay. Mm-hmm. Yeah, this was good. It was very good.
No, I like this.
It's a TWiV open forum.
Okay.
Mm-hmm. Like it.
You can find this, of course, on iTunes at twiv.tv. And if you like us, go over there on iTunes and rate the show or leave a comment. That helps to keep us visible so we get more subscribers. And, of course, send your questions and comments to TWiV at twiv.tv. Dickson Despommier is at verticalfarm.com, Racaniello.org, and medicalecology.com. Thanks for joining us, Dickson.
It's my extreme pleasure.
Don't you love how your websites just roll off my tongue?
I do.
Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
Sure enough. Always a good time. Good fun.
And Alan Dove is at alandove.com and on Twitter. Thank you, Alan.
It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.